I asked you all to fill the survey form, fill up, and we got quite some response. So I'll cover that. Then we'll look at the why do we need simulations at all and MD specifically. And then we'll go through the steps. So what is needed for minimal setup of the model, force fields, a bit more in detail, and the algorithm of MD. And then we'll take a break. So I think it is now 12, but the trend is still the same. So the survey showed that most of you have at least some bits of theory, and that to some extent explains why you are here. So I guess you are interested to some extent, you know something, but not many had it as a part of real project. But more than half wants to, or yeah, people in general on average want to consider it at least as a part of their future project. So that is quite promising. And we got just a few tips on what some of you want to study or working on. And at least part of it we will cover during the lectures, and some will be taken at the end. So in this last questions and answers session. Yes. So first of all, why do we need computer simulations at all? And specifically in the pharmaceutical field and some adjacent fields, because we want to study large biological systems such as membrane or protein in lipid environments, maybe some drug solubilization and some similar systems. But we want not only to see what is the most most relevant statistically equilibrated system, but see how it evolves. So that is something on that time scale, on that length scale, that is not really possible to observe from experiment. And also it can be quite cheaper, relatively cheap, relatively fast, and we can really sort of preserve all the variables except something that we want to change. So as compared to experiment to the lab, it might be a bit harder to control the environment completely. And then we can use it either to predict what would happen in the biological experiment or to explain what already happened in a biological experiment if we try to clarify it. But clearly there are some limitations of the approach. So we can only target, specifically with classical molecular dynamics, we can only target the length scale range between nanometers and maybe several hundred nanometers. And the time scale of maybe tens of nanoseconds to tens of microseconds, so around that time scale. These two are basically limited by the computational resources that would be needed to simulate such systems and how well we can parallelize this box. Another is that we cannot really increase time step because there are physical limitations such as the frequency of the hydrogen atom oscillations. And then if we go above 2 femtoseconds, yes, then we won't really preserve this carefully enough, and it won't be really good simulation anymore. But at higher levels, so if we cross grain, of course, it would be a bit different. The force fields themselves, I will cover that a bit more specifically, are not perfectly accurate, and covalent bonds are not really getting broken or formed during the simulation, and electrons are ignored. For that, we would need a quantum mechanics calculation, and I mean, yeah, QM simulations. So keeping that in mind, we can actually just briefly cover a few of those applications that are very prominent at the moment. So protein folding pathways, so this funnel-like shape was discovered, and I mean that's quite a lot of actual papers around that. Ligand receptor binding, so we can find the most prominent ligands and inhibitors, and so quite a number of applications. And the membrane molecules interactions, so either those complex membranes with proteins incorporated or something simple. We can really study interactions with a number of external media and colloids and intestinal fluids and so on. And On the top of that, it's a core of many solubility studies, or at least solubilization and self-assembly of those colloids that I mentioned already, optimal formulations with free energy calculations, and many, many others. Well, some of them we will cover during the sessions today. But to solve any of this aforementioned problem, we need to start with particles. So all of these systems contain atoms and molecules. So we need to introduce those. And if we take a simple one, an argon atom, and if we introduce it as a single bit, then there would be at least few rules that we would definitely need to follow. First of all, argon atoms wouldn't really overlap too much. I mean, even their electronic clouds wouldn't really overlap because they would start to repel each other quite significantly. So that should be taken into account. At the same time, if the atoms are way too far from each other, so the distance between the two centers of masses or geometrical centers of these molecules is much bigger than the sum of the radii, then there will be no interaction. We won't calculate nothing and use our computational resources for that. And at the same time, there might be some attractive effects right between the atoms, because there could be, for instance, induced dipoles, and then these two atoms could stick, or atoms could stick together because of that. And so to cover all of those three, we could introduce first Lennard-Jones potential. So basically it's van der Waals interactions we're trying to mimic with that. And that would already include the repulsive part. This red line here shows the, as you can see, positive potential. So that's not where the two atoms or molecules will be happy to be at this distance from each other. The blue one show the negative values. So the attraction, as in the case of induced dipoles. And so if we sum those together, we will get this profile, which is very nicely defined by Lennard-Jones potential. So this one is a classical form. I think it's the most common, 12.6. So this first term here defines the repulsive part, that one. And the second term is responsible or stands for this attractive part. And this epsilon in front is the strength of that interaction. So basically, this repulsive part, it will be super strong in any case. So whatever, more or less whatever you have for epsilon here. But this attractive depends on it quite well. So basically, by setting up how deep is that well, so how much is that difference, we will set up how much of attraction will the molecules have between themselves. And in that way, if we still set up these values for all pairs of groups of atoms or atoms, then we can actually mimic the real system. It's just another illustration of the same concept. So there's a repulsive part and the attractive part, and this one is very steep. And basically, they stand for this electronic cloud overlaps. So once they start overlapping, that's already a repulsion. That's how we mimic this real system. So this is a very common model for that sort of interaction. Apart from that, it is important to take into account the charge, so electrostatic interactions. And for a simple case of two charged atoms of sodium and chloride, we would just use this Coulomb-like potential, which depends on the two charge and on the distance between the two. And of course, in reality, so this one, the main difference between, well, maybe not the main, one of the differences between the two potentials is that this one is much longer scale interaction. So technically, that should be really across the whole box, that the particles can feel each other to some small extent. But in reality, in most of the simulations, we use some cutoff as well, in a way, to limit the number of calculations we need to not just calculate it for each pair of atoms, whatever the size of the box we have. But other than that, it's quite simple here. A bit more complex case is when we have when we need to introduce partial charge. So if you look at the water molecule, you can see that actually it's polar. So though it's overall neutral, still it would be it's polarizable. So technically, we need to take care of that charge distribution across the molecule. And to do that, there is also quite big subfield of the entire theory of molecular dynamic simulations with the partial charge. So that's one of the important steps we need to go through once we validate and test our model of a molecule. And just as an example of the water molecules, there's quite a lot of those. So for different types of simulations, for different systems that we want to mimic or model, there are, I think it's like hundreds of water models that already published. And basically, the more complex we go, the more expensive it becomes. So then for each molecule, we would really need to spend more time to find out how it would interact with others. But there's an optima. So the most common that we, I think, use is tip 3. So that allows to define this partial charge, but at the same time, it's not overly expensive. There's quite a number of those. So yeah, if we have a charge, if we have these attractive repulsive interactions, it's still not everything, because not all the molecules we simulate are ions or single atoms, right? So those make up molecules. And in order to simulate molecules, we need to bond those atoms. And as you maybe remember from one of the previous slides, there are no covalent bonds being formed or broken during the simulation. So we need to set up some interactions between those to make it a single molecule. And normally it's done with a spring bead model. So this, let's say, atom has all its interactions with the rest via electrostatic and van der Waals, but at the same time, it's connected to another one with a spring. So sometimes it might be also rigid bonds, so constraints, which limits it much stricter. So basically, this one is more flexible and maybe a bit further away from reality in a sense. But on the other hand, that one would cause much more troubles with the simulation being broken. But again, that's a bit specific to a problem that we want to study. That is something to take into account as well. And this spring-like model is normally just set up with using the Hooke's law. So there is this optimal position, optimal distance between the two atoms, which is L in that case, this reference bond length. And then this K defines how strongly are they pulled or pushed away from that position to reach that position from the current position R, which was caused by all the rest of the potentials and interactions with other molecules. So that is not optimal, this harmonic potential with the spring. So there's, for instance, this Morse potential that is much more accurate, but it is more expensive. And that one is shown to be close enough. And so again, if we look for optima to run something not within years, then maybe harmonic potential is a better choice, at least to start with. Another important thing is that we do not just need to connect these atoms in any way, but we need to also preserve the angle. So let's say for the water molecule, if I'm not mistaken, it's 118 or 120 degrees here that need to be preserved to simulate water properly. And that is also introduced via this simple model. So we have the strength of interactions of this potential defined by K, and the optimal angle and the current angle, and that is the difference is squared. So it will try to preserve it in that shape. And then there are also dihedrals. So I have some videos, maybe that is better to illustrate. So basically, it's like with this arm model. So if we have as a reference this CC, CB, CD segment that is stationary, that one could be entire segment that could be rotated around that. So we want to preserve, so as you can see, this angle here remains, let's say 120 degrees, but at the same time, it still should not rotate around its axis all the time, at least for some molecules, right? So that is one type of dihedrals, and there is another one called improper. So this is when you have the reference of the CD, CB, CA, and CC. So this whole thing should be, so this hydrogen atom should be stationary with respect to these four. So those two are again important to take care. Otherwise, the molecules will be just overly flexible and they will perform the behavior that we wouldn't see in experiment, or actually that doesn't exist in reality. So there are limitations and we need to just set those up. Okay, and then if we just summarize all those, if we look at the molecule of some small drug molecule, I think in that case danazole, in water, we would of course need a whole bunch of these interactions. So each of these atoms should be correctly interacting with the water molecules, with the surrounding molecules. Normally there are exclusions, but this is a bit too advanced maybe now. Yeah, so the interactions should be set up properly. And I mean, we also could have other molecules of that kind or some more other type of molecules. So each of those should be set up. Bonds should be set up, angular potentials, dihedrals, and altogether, when we describe all these interactions, that altogether make a force field. So the description of all the interactions between all types of atoms, whether they're bonded or not. And that's something that we want to get to now. So there are at least three groups of force fields that I would like to mention here. Is all atoms. So when we really introduce each atom and even hydrogen atoms, united atom is a bit more higher scale. So basically we then substitute, if I'm not mistaken, again, there are a few modifications, but let's say all hydrogen atoms are said to be a part of a heavier atom. So in that way, we can increase the time step, which I mentioned that it limits our simulation to have only 2 femtoseconds per step. And then we can just reduce the number of particles that we need to calculate. And so it makes it faster. And then there's coarse grain. And Shakolat will cover more of that. But yeah, in 2 words, it's just that we take even few heavy atoms to make one beat. And those beats represent, so they still can be able to represent the entire system, but just with a much fewer particles. But if we talk about all atoms, then charm, bromas, and amber, I would say, and maybe OPLS would be the most used, the most common ones, and the ones that I would recommend to start with, at least to read about those. There's quite a number of differences. And maybe just to mention that charm is rather a bit more of a empirically derived, so from experimental data, whereas the amber and bromas are more ab initio based. So from first principles, those are derived from physics that we know. But yes, so as I mentioned before, there are many force fields, and they kind of combine, make these parameters, interactions, all pre-written for us to run this MD. But we always need to keep in mind that, first of all, they're quite simplistic models, so they do not really fully mimic the behavior of real systems. And then they are derived in such a way that they would be applicable to some specific system, to some specific task, basically, to address specific tasks. So we should really carefully choose one. Yeah, and the development is quite active all the time with force fields. So maybe one day it will be an optimal that will cover all the problems at once. But for now, if you want to choose a force field, this actually was one of the questions in the survey. Maybe it's a good idea to start with resolution. So whether you need all atom or you can go coarse grain. But for coarse grain for validation, quite often you would also need all atom. And then you would really need to think about the purpose. So what exactly you want to simulate and how you want to simulate it. And then maybe you would need to read the force field descriptions, as for instance on the GROMACS website. I will get back to GROMACS a bit later. But here you can see, so down this link, you can see the description of all, or maybe not all, but numerous force fields, even subversions. Yeah, so basically,
I think it's fine with it. No.
Yeah, I think
yes.
we are
Let's try to say
with someone. Yeah. So yes, basically each of these versions might be optimal for some specific.
It's just the
problem.
number they want.
Yeah, yes. I think we need to mute one of the participants.
Is that a Finnish name?
Okay, so yes, and finally, or maybe not finally, one of the best cases to even first thoughts to take into account is the similar studies. So if you can look into the similar studies that were published, and if they're well cited, then maybe that's how people normally do that. That's the force field that is commonly used for that kind of problems. Yes, but let's say we have chosen the force field. We understand how to set up interactions. So how do we actually run the MD itself? And that is where the molecular dynamics step in. So this method systematically solves the Newton's second law. And if we either randomly or not randomly put the molecules in a box first, and then we start with minimizing the box. Once there are no overlaps, we start really solving the second Newton's law equation, more or less iteratively. So we just solve this equation based on the potential. So if we have this force field defined as we had it before, this U total, right? That would summarize, if we take the sum of all the particles within the cutoff distances, we will know what's the total potential acting, or on that particle in that point of the box. And then if we just from that derive the force, and we take some specific time step, we will see where the particle will move within one step. So from potential, we find the force. Force leads to acceleration. Acceleration with time together gives us the where this particle will move at the next cycle, within one cycle. So just to illustrate it, imagine that we have these two limits, and they're not really in vacuum, but there is maybe water around and some other molecules. But if these two are far enough from another, then there won't be a lot of interactions happening. I mean, if the cutoff distances are not overlapping. But there is, apart from conservative forces, this potential that we described before, there is also friction, so dissipative force, and the random force. And random force comes normally from thermostat, which again we will cover later. But that might be inducing these thermal fluctuations that can, via either random motion or via some other molecules, push these two towards each other, for instance. And if it happens, then they will start seeing each other, and then conservative force steps in again. So that will be van der Waals, electrostatic, and so on. Basically just those two. There are no bonds here. And then we will see already a bit different behavior. So they might be attracted to each other, or if there's no attraction, they can just be repelled. And not really repelled, they just will ignore each other in a way, so they won't overlap at all. So that's one time step. And then we calculate it again. So in that case, there were some other molecules that push them together. On the next step, we already calculate also these interactions and all the rest. And then we calculate what will be the next one, where will they end up in one delta t time. And this is basically the algorithm. So we start from initial configuration, then we calculate the new forces from these potentials. We solve the equation of motion to find where it would end up in one cycle. And we write down the position and velocities, forces, and we go back to calculating the new forces. So basically, we do it in a loop for the entire trajectory, for let's say 2 microseconds or whatever it is. This is just a simple example of how it could look for a water molecule. So it just moves with this 2 femtosecond step, bit by bit. So it could be either due to thermal fluctuations or interactions with other molecules, but it just it doesn't stand on the same place. But as you can imagine, we don't really run a lot of simulations in vacuum. So there should be the box, and the box should not be empty. So it should be filled with either water or some other solvent, for instance. And again, we cannot really set up we can, but in most of cases, there is no point in setting up the simulation so that there are strict boundaries where the molecules would bounce back in MD. That's why one of the important things here is periodic boundary conditions. So once the molecule comes to the side of the box, which we define, or yeah, again, it depends on the barostat as well, which I will come to in one slide, I think. Once the molecule comes to the side of the box, to the edge, or so, yeah, it can, on the next one, jump across the boundary from the other side. So basically, it's like you're simulating just the central image, but it can go through each of the sides to the central image again on the next step. So yeah, it's just like a teleport from one side to another. And the important thing here to have to take into account again is that your interactions, so your box should be big enough to be more than the distance, this cutoff distance of the atoms, so that it wouldn't feel itself from the other side if you use periodic boundary conditions. Another rule here is a bit maybe less conventional, is that if you have, let's say, a colloid in your system, in your box, which you simulate, so basically it occupies a huge part of your box, you maybe should have enough solvent so that the colloid would take less than half of the length of the box in any direction. So this is sort of a similar approach. It won't explicitly fill itself, but it can through the, let's say, solvent. So it kind of, it induces the other molecules to change behavior. And so it can be self-consistent in that way. So we want to avoid that. So that's why we need to run relatively big simulations sometimes, boxes. So this is just another example of the box of water molecules that are all interacting with each other. I think it's the same time step of 2 femtoseconds, or maybe bigger. But yeah, and that is how, let's say, simple case box could look like. And if the molecule comes to that side, on the next step, it could appear from the other one. Yeah, to control temperature, so we would need to have a thermostat to connect our system to a thermal bath in a way. And then there are 3 typical thermodynamic ensembles which set up the rules. I mean, there are more than that, but these are the most common ones. Microcanonical, where you preserve number of particles, volume, and energy of the system. Well, you can imagine that if you want to preserve number of particles on volume, that's sort of a closed box. So for some experiments, that would be maybe the best match. Canonical is where you preserve number of particles, volume, and temperature. So this is maybe closer already to real experiments. And then you just keep the, like for instance, in unfolding of proteins, it's quite important to keep the temperature at a certain level. And maybe the most common one is isothermal isobaric ensemble, so where you preserve number of particles, pressure, and temperature, so not the volume. So the box size can vary to preserve the pressure. And that is, I think, most relevant to a majority of experiments, because we have, let's say, 1 bar atmosphere, and we have either room temperature or body temperature, depending on the process we're simulating. But yeah, that I think should be the most common case. And in order to control those, we basically do it either via energy of the molecules. So we scale up velocities or scale down velocities depending on the temperature at the moment. And with the barostat, it's the same. So if the pressure is becoming too high, then we just expand the box a bit. And I mean, it's all treated with quite a number of those barostats and thermostats. Yeah, and I'll just wrap up now soon with the first session. So, and all these rules, so MD algorithm and quite often force fields are already incorporated into some of those software packages. So maybe the most common one is GROMACS, but there are also many others like Amber, Charm, Desmond, Express MD. And many of them are based on their own force fields, such as Amber and Charm. GROMACS rather can use all of those. Well, at least as far as I remember, all of those here: Amber, Charm, OPLS, lipids. And yeah, most of those are freely available to academia. I just maybe will cover a few of the advantages of GROMACS, because first of all, it's user-friendly. There's a huge community, a huge number of advice you can get over internet. It's quite well balanced in its code. I mean, it has a very high performance, and it supports GPU impairment, parallelization. So you can actually make your simulation much faster via the tools that are already there in the GROMACS. And then, there's quite a number of other advantages as well. So just to very quickly cover the advantage of GPU and parallel computing, this is from GROMACS website. You can also read more about that. First of all, if you use graphic card, powerful graphic card, it might increase the simulation by orders of magnitude, basically, specifically if your system has a lot of small water molecules. So basically, many small molecules are much better treated with graphic cards if you have one. And at the same time, it's a pretty good exercise. So it's like a small tip here to start any simulation with running some relatively small interval of time, period of time, at different number of cores. And it might happen that you find the optima, and then let's say you find out that with 8 cores it is the fastest, and let's say it's 2 times faster than 4 cores, but with 16 cores it's more or less the same or a bit slightly higher than at 8 cores. So in that case, you would better run 2 parallel simulations with 8 cores each than running it at 16. So this is something useful to do, but there are a lot of studies on that, so maybe you can also look into those. So in the second part of that session, I will cover the basic measurables, basically, that you can evaluate from MD simulations, and then we will stop a bit more properly on the free energy calculations and some examples on RDF, number of contacts, hydrogen bonds. And then I will summarize take-home messages before we go to a bit more practical part of our session. So first, with the static properties, so the ones that are measured at equilibrium, those are, if we really start from scratch, from simple ones, those would be kinetic energy and temperature that are, of course, interrelated. So from all the velocities that you, of course, can track along the trajectory along your simulation, you can find out what's the average kinetic energy. And in such way, of course, you can also control and track the temperature, which might be very important, as we mentioned already, for proteins and some other sort of simulations in biomolecules. Yes, so that's something that is commonly very easily measurable in the simulation. Configuration energy or total energy maybe of the entire simulation is quite common as well, to see whether the system is equilibrated, whether it's the volume, it shrunk to the proper state, which can also be done evaluated with the pressure. So you can see whether it reached the plateau. And in that way, again, you might need to control pressure in some more advanced setup if you're mimicking some industrial or process. And specific heat as well, that can be calculated basically via the same measurables in a way. A bit more interesting, a bit more practical, I would say, is the pair correlation or radial distribution function. So that I will show on an example soon, and that is closely related to structure factor. So basically how the atoms are organized or molecules are organized within the simulation box, if they're forming some aggregates or not, if they're evenly distributed or there's some trend within. So more specifically, radial distribution function shows how are molecules distributed with respect to some specific type of molecule. So let's say if we want to study we have one water molecule here sitting in the center, and we want to see how the other water molecules are distributed with respect to it. So then we set up a bin width, so this sort of a ring, so how wide it is, and then we start from the first, from the closest surface of this molecule, and then we increase it, and we calculate how many times, how many atoms have their centers of mass, for instance, within those circles. And then, again, if we have the system calibrated, that would already give you some profile. You would see whether there are some inheterogeneities or inhomogeneities in that profile, which could be quite often compared to experimental data. So this is a typical RDF for such system as here. So the first layer would be here. So so before that, there's just no possibility that the centers of mass of one molecule or another can be closer to each other. Again, that would be an overlap of this, for example, electronic clouds of the atoms. But then, after the first formulation shell, there's a well, because again, there is this gap just on the next generation, and so it smoothens out with this new generations, or with further distance from the original molecule. And so that is calculated for each of those. And you can imagine that these statistics accumulate pretty fast. So for each of the water molecules, you try to see how many other water molecules or any other type of molecules are at which distances from it. And again, that could be pretty well compared. So that RDF, radial distribution function, could be very easily converted with mathematical tool to structure factor, something that you already can measure with the stacks, for instance. So those peaks would characterize what are the typical distances between some atoms within the sample. And knowing that, you already can draw conclusions how it's organized within your sample. But the structure factor itself, you can also calculate from a molecular dynamic simulation. So you don't really need to go through RDF. So RDF is just a bit more intuitive by itself. So if you don't compare it with experimental data, that is a pretty good tool to see what is happening in your system or compare some systems. I can show you an example. So let's say we have a system with, so this is meant to be amorphous solid dispersion, but in the presence of water. So we add some humidity here. So let's say we have a polymer that is supposed to stabilize the drugs, prevent them from aggregating and crystallizing and nucleating and crystallizing. And there is also water. Water is blue, polymer is yellow, and the drugs are, water is red and white. Yellow is the polymer and blue is the drug molecule. So a bit counterintuitive colors, but yeah, I think I just wanted to keep the colors from the previous one. And for one drug, which is more hydrophobic, you can see that with time, it actually cells or phase separates. So it becomes a mixture of polymer and drug, a mixture of polymer and water, but the drug, the blue part, is separated. So it doesn't really allow many of the water molecules to be incorporated in that. And that is another case with a more hydrophilic drug. So at least there are some parts of it. And as you can see, so it's more or less all the same, except that it's another point of view. But again, from another point of view, that would be the same. They are much more properly mixed within that system, all the drug, polymer, and water molecules. And to see that, you don't really need to, because we need to quantify it, right? Not just visual observe it from the simulation. RDF would be one of the tools. So if you look at that graph, specifically for that system, so drug 1 and 3 should correspond to those two systems. So, well, actually, yeah, I wish we had time. We could have a quick check if you could understand which one is for which, but I think maybe we will skip that. So if you look at the interactions between, let's say, API and water, so for the first one, as you can guess, the interactions would be less pronounced. So the chance that the profile would be around 1. So 1 is like unbiased. So basically, there are no difference between being next to it or not. So it's like equally good or bad interaction. If it's below 1, it means that there is a lower chance that these two molecule types, like API and water, for instance, will be found at that distance. So it's lower than 1. That means that they really not prefer those kind of interactions. So APIs in all cases actually in that case, it was 3 drugs, but I just dropped it to 2. They really like itself the most. So these are just limitations, geometrical limitations of the drugs themselves that cause these oscillations. But as you can see, it's everywhere above 1. So which means that it would self-aggregate in any case if it has chance to do so. So one of them, if you look at the green lines, one of them has a lower profile for interactions with water, and another one has a higher one. And you can guess that the solid line corresponds to the first one, so where it likes to interact with water less. And the other one, which was more or less properly mixed, it almost reached 1 at the short distances. So already that is a way to quantify the interactions taking place in your system. And this is just an illustration of the same. So so this is API number 3. It's more or less mixed. So only the API molecules are shown here, and most of the white space would be water and polymer, but yeah, those are mixed. Here it rather aggregates and tries to make one colloid. It's just not a completely finalized simulation. Otherwise, it would really make one. So that is one very important tool, and I think it's used in maybe 50% of all the studies around MD. Another one that would be good is the numbers of contacts that you can count. So it's sort of a similar message that you could extract from that, but a bit differently. So then you just look at the real neighbors of that particle, whether they can still see each other, and then you can count those. And this is an example for these drugs 1, 2, and 3, these columns correspondingly, and how more preferable are, let's say, interactions with itself than with water or polymer. And at the lower drug loading, you can see that the drug 3 has approximately the same number of contacts with polymer and with itself, and quite a few with water. Whereas for the first one with this aggregation, it's mostly with itself, and very few with waters. So, and with the same tool for instance, in GROMACS that I already sort of advertised in a way, you can also measure the minimal distances between the atoms of one type and another one. So they kind of go in line. How many molecules are at which distances? So it's quite common to use correlation functions to see whether some parameters depend on others, and to explain what actually happens, what's the cause of some specific phenomena in your simulations. That's quite common. And another dynamic property of interest is mean square displacement. So that one basically shows how far the molecule, so basically it's diffusivity in a way here, right? It's how far the molecule travels within a period of time from its original position. So if let's say you take as a reference a protein, and there are drugs that, that can be sitting on the surface of that protein, either it can stay there or it might just travel away, and that you can evaluate. And from that, again, you can draw conclusions whether it binds or not. Maybe drug and protein is not the best example, but still the message is the same. That's quite a good tool as well. And diffusion coefficient. So and another important way to use it is to understand the phase at which this molecule is currently being. So whether it's solid or liquid. So the liquid, it would drift away gradually. I mean, if the box is big enough, but in solid, it would be oscillating around the same spot. And actually, there is also two other that I don't have in the presentation here. It's RMSD and RMSF. So root mean square displacement and root mean square fluctuation. So those are slightly different. So fluctuations, for instance, it's mostly used for parts of the protein to see whether those are oscillating more or less and how it changes with time. And let's say with that, you can see whether the protein unfolds. And yeah, so these are some of the tools. And as in many of these equations that I just showed you, there were averages. It is important to mention here that average can be taken in two ways. So it's either time average or ensemble average. So one is when you just run the simulation long enough, and then you maybe can observe all possible configurations, all possible positions and interactions. And another one, when you run many parallel simulations, and when you start from different configurations, from initial positions, let's say. So technically, if you have a very long simulation and very big number of these ensembles or initial configurations, those should be, the effect should be equal. So this is the ergodic hypothesis that the time average is equal to ensemble average. But as a rule of thumb, I would say it's better always to use at least 3 simulations, so triplicate, run it in triplicate, and for long enough time to see whether they are consistent with each other. Yes. Another big field, I would say, is the free energy calculations. I think it's not only limited to MD, but it is a big part of it as well. So that can tell us quite a lot. So if we look at the Gibbs free energy, that can tell us whether the reaction, so whether we have observed any change in the system, whether that reaction would be spontaneous, whether it would happen by itself in the box. Yes, and that could be applied to quite a number of different tasks or different questions to be addressed. So for instance, solubility, whether the drug would be better soluble in another solvent as compared to, let's say, water. And then whether it would go to the membrane, or whether it would penetrate or stay at the interface, or to compare different drugs, which of them would bind to protein the best. So in order to evaluate those with free energy, we would need to really cover the entire space of configurations. So we would need to see how likely the molecule would be found in one state and another, and then to measure it together with the probability of finding this molecule in a specific state. So basically, that's what we multiplying here, the Hamiltonian. So the description of the system in terms of the positions of the molecules and their momentums, and the Boltzmann factor. So the probability, how likely it is that the molecule will be found in that state. And when we put it together, we can compare the different states of the system. And I will give you a few examples now in just 1 second, by just subtracting one from another. So we never really look for absolute energy, free energy. We always look at the comparison of those two. And then this is basically the same as just comparing probabilities of these two states of the system. So one simple example would be if you have a solvent and if you have two slightly different molecules, as you can see here. If you just run free energy calculations with few of the possible techniques, I think we won't really go into details with that. It's like thermodynamic integration, it's free energy perturbation, many others, Bennett acceptance ratio. So those could be applied to evaluate how much energy would be released or would be needed to, let's say, delete that molecule from the system full of a solvent, for instance. So that's one of the ways. That's with varying the lambda. But again, I won't really go into details. But if we compare that one with another molecule, which is slightly different, and we will see how much energy change would be between those two, between deleting one from the system full of water, let's say, and another. We will see how more likely that molecule would be soluble in that box, let's say in water. So this is already a comparison among the different molecules. Another quite common thing you could see in publications with MD and free energy calculations would be free energy profiles. And I think you saw some of them. So let's say if it's a membrane, and that would be looking something like that. So, and to interpret it, I have an example with the rock concert. So just imagine that there's a rock concert and there's a lot of people, and there are a lot of different kind of people there in this audience. Some are really big fans and they want to stick as close as possible to the scene. Some are having this, I'm not really sure I'm using the right terms, but this mess in the center. And if we just show their happiness at different places with the color, or maybe their activity, I would say, you could see it on that map. So there's a spot of those having fun in the center, and there's a lot of those dedicated fans in the front row. The blue ones are a bit more moderate. So yeah, just to again illustrate what kind of audiences that could be, right? So, and then if you compare that to the MD simulation, that would actually make some sense. So there are molecules that would prefer to stay in the quieter place. There are molecules that would really try to get as close as possible. So in the case of the rock concert, that would be the biggest fan that is, so it's a bit counterintuitive here with the axis. So happiness goes, I mean, the happier, the lower here, the lower, the happier. So far away, the biggest fan is not happy, but it is happy at the first row, and maybe would be happy also on the stage. But there's a security line that prevents biggest fans to go to the rock band. And there are moderate fans that are quite fine with that, and they just don't want to get into mess, so they are staying a bit outside. And they definitely don't want to mess with the guards and with the rock band itself, so they are just listening to the music. So it can be interpreted the same way with the molecules and how happily they would go inside the membrane. So if you have a lipophilic molecule, it would maybe be the happiest, if I can say so, at the interface, where the hydrophobic part would stay within the membrane, and the hydrophilic part would stay outside in the solvent, in the water here. Whereas if you have a water molecule itself, it definitely doesn't want to go inside the membrane, so it would stay outside. And then here it's equally fine elsewhere, right? But once it's approached the membrane, it becomes less happy. Or the negative free energy here would be lower, right? So basically, I'm sorry if I'm confusing you with the sign here, but the lower the value of free energy, the happier the molecule it is in that specific place. And then with that, again, you can study permeability. So how well molecules would, how would they interact with the membrane, let's say, or micelle, any colloid, how well it would be solubilized within some media. And the solubility you can measure as well with free energy, but it's, as I mentioned, it's not absolute solubility, it's just mostly in comparison. So you can just put different molecules into the same solvent and compare free energy changes, or you can put the same molecule into different solvents and again see in which solvent it's happy. At least qualitatively, you definitely can do that, but quite a number of studies show it pretty well comparison with the experimental data. So many of those are very reliable. Evaluate with that combination, solubilization, stability, you can evaluate the efficiency of enabling formulation. So how happy the molecule would be in the presence of that as compared to, let's say, water or intestinal fluid. But to simulate that, this is another question that we're addressing from the survey. That would be enhanced sampling. So we need to fix the molecule, because if it wants to go to the membrane, let's say, or at the interface, we need to really collect enough statistics at all the states, not only at the interface, but within the membrane, outside the membrane, and so on. And it only wants to go to the interface. So we need to restrict it. So that's something that is called enhanced sampling. So we fix some of the degrees of freedom of that system to collect a statistic in that space, because otherwise we will really have some just pieces that don't really give us the entire picture, don't give us the free energy profile. So, and if in case we want to pull the hydrophobic molecule away from the micelle, we would need to, well, first we would need to have a equilibrated system with the molecule in the center of the micelle, and then we start pulling it away. And at each of these, so we take this trajectory and each of those slides, or some of those slides, we save these configurations when the molecule is here, here, and so on, is as compared to, or with respect to the center of the colloid. Fix it, and then we're gonna fix this degree of freedom. So this molecule is only allowed to be, let's say, within that sphere. So it cannot get closer or further away from the micelle surface. So it's either fixed at the surface, within, at some of the shells, or outside of it, with a spring potential as well. And then once we run all these separate configurations, once they can only get around this at some certain distance, we can then pull them all together, and with something that is called weighted histograms method, or maybe there are also alternatives, but again, I won't go into details. You can put it all together and get the actual energy profile, which here is in kBt units, but yeah, it could be also in kilocal or whatever. So, and then you see how happier the molecule is in the center or inside the micelle. So let's say in that case, it would be something like 2 or 3 within and 20 outside. So it's very likely to go in the center and be completely covered, shielded, screened from the water, contact with water. That is just one way. This is called umbrella sampling. So we just cover all possible configurations and run those separately in parallel normally. But there are basically many, but the two most common analogs maybe are metadynamics. So again, if there are barriers, if there are states where the molecule is not likely to stay, so let's say it's happy in the center of micelle, or at some inner shell, and it's happy at the interface, but a bit away to solvent, but not between the two. So it's hard to simulate that specific spot. And then to address that, there is this artificial extra potential, if I can say so, added, which fills those wells. So we simulate it in the wells, and then we sort of balance them with extra potential so that the molecule would not really get into that deep well. So it would stay, it would just jump around, but it would be more likely to be found in that intermediate step. And then you increase it even more, and then you just cover, like you fill it with water in a way, of extra potential, of extra free energy in that case, to go to all possible states. And another one is, so this is like alchemical simulation. So we really implement something that wouldn't be realistic in real world, but that is still representing an important system. And then with replica exchange, it's just that we run several configurations in parallel, and then we exchange the temperature or configurations, and to see basically, well, to just go to prevent them getting stuck to that local well. All right, I'll just very quickly go through another example here. So that's how we applied this with some free energy calculation. So we wanted to see whether the molecule can be transported with the micelle. So let's say if it already solubilized, got solubilized within the micelle, would it really, this micelle is bile salt and phospholipid, would it really be delivered or shuttled to the membrane, and would it then leave the micelle? Because otherwise it would aggregate and form some cluster that wouldn't really penetrate the membrane. So, and two potential good scenarios would be that it would just give away the molecule, or that the whole micelle would join. In both cases, we can study with free energy calculations. So this is something we published actually also together with Shagavat. So we just found out how more happy, if we use the same term, the drug molecule danazol in that case would be sitting as compared to water within the membrane. You can see that level here, and within the micelle, and that there could be a barrier between them. But if the micelle is at the surface of the membrane, then it's more likely to just overcome this barrier to go to a bit slightly happier place to the membrane, as compared to if it needs to leave the micelle and then go to the membrane just as a single monomer, as a single molecule. So that means that this micelle can serve as a shuttle bus for that molecule, for some of the hydrophobic molecules. Another thing that we can measure, which is also important, is hydrogen bonds. So that is, I think, now also introduced in some of the coarse-grained force field, but is mostly studied up to now in all atom simulations. So this is just an example of, again, polymer that stabilizes the drug, and then you can just evaluate so basically how stable the hydrogen bonds that have been formed between the API molecules and polymer, or like here between API and protein. And then what are those specific amino acids that form those bonds? So, and from that, you can come up with a whole bunch of recipes. So how to improve whether the stabilization algorithm for these drugs or something else. So hydrogen bond can be used in quite a big range of different projects. So that is something, again, that could be analyzed with the molecular dynamics simulation. So, but I'll just start to wrapping up here. And first, I wanted to mention again that the simulation do not really represent real systems. So it can, if you do everything carefully, it can give you some insight, but just don't take it as a pure truth straight away. And everything should be studied with enough statistics. So if you just observe some, let's say, passage of the molecule from one media to another, or transfer from micelle to membrane, it doesn't really mean anything. It can be some eventual simulation or event, But if you observe it either with enhanced sampling, or if you really observe it in numerous of those simulations, it just as a trend it happens, then that already might mean that, okay, it might happen if everything is correct with parameters, it might happen in reality in the experiment. And it's always good to try to estimate the errors that could be accumulated in your simulation. So apart from MD molecular dynamics as such, there are quite a number of other metascale models, so which address the same scale, the same time scale, the same spatial scale. That would be a dissipative particle dynamics. So the principle there is the same, it's just that we substitute the very hard repulsive potential, you remember that steep slope, with a more smooth one. So we kind of shift it all to the right, but make it smoother. And then all the particles are softer, and it's rather how well they attract each other. But yeah, the principle is the same. And then there are other methods, let's say like Lattice-Boltzmann method. It by itself, it doesn't really represent the same, but it could be merged. It could be used it could be coupled to MD, but that would, let's say, replace the explicit solvent. So then you just use another approach to simulate all the water, for instance, and that will be much faster because in majority of the problems, water takes most of the computational time. And the main problem in all of those methods is to set up the interactions and parameters correctly. So you need to validate it with some experimental data, and that is one of the main take-home messages here. So first, it might be hard to study with experimental methods, then you can apply MD. So there is a gap between experiments that might be quite well filled with molecular dynamics simulations. But of course, it has limitations that we already mentioned several times, and it most certainly needs to be validated with experiments. So you need to, or it should be very well based on the ab initio principle. So you need to be sure that your model is good enough. And if you want to start using MD, there are lots of software packages such as Chromax and others that already have lots of pre-written code. So you will just combine the commands instead of writing the whole algorithm by yourself.